I think there's a lot of opportunity with this vesicle technology because they can be delivered through the skin. I think in terms of cosmetics and skin diseases, there may be opportunity there. They also can be delivered orally. So like I mentioned earlier, delivering these antibodies orally or other immunotherapeutics orally, I think would be pretty neat to do. All this is going to take clinical development work. And right now we have a lot of ideas that we need to prioritize and just bring one product at a time to the clinic.
Building a novel delivery platform is one thing. Manufacturing under GMP with no established playbook is another challenge entirely. Christopher Locher returns to continue our conversation, taking us deeper into the practical realities of bringing a first-in-class biologic from bench to clinic. The infrastructure decisions, the analytics, the regulatory conversations that don't follow a template. Christopher is the CEO and co-founder of Versatope Therapeutics. He has navigated this path from multiple angles across his career. Let's continue. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. What is your company approach? You mentioned the word CDMO. As a smaller company, you always have the challenge of what do you focus on? What do you outsource? What do you insource? What is your take there?
So one has to have a pretty firm understanding of what they have and how to characterize it before they tech transfer it. So we develop all our assays in-house here in Massachusetts before we sent them out to CDMOs for tech transfer. We made the decisions on some products to outsource because we didn't have the equipment to begin with. But once we got the equipment, then we were able to do that work ourselves, optimize the test, understanding what the parameters are, the standard deviations, and then able to tech transfer it in a robust template and format protocol that then can be used at the CDMO or wherever it needs to be manufactured. We also did a lot of our own analytical tests. We have 24 qualified assays. We didn't qualify all of them in-house. We qualified some of them, but they were in a position to be qualified with the right people and the right staff at a CDMO once we had those protocols in place.
And did you consider working with CROs as well for analytical as well? Or that was not part of the strategy because you developed everything in-house?
We've subcontracted some work to those folks, and our CDMO has subcontracted some work, not a lot. We prefer to do the bulk of it with a single one-stop shop to do the manufacturing and the analytics in one go. That includes both drug substance and drug product. I'm not a big fan of shipping stuff around, and if we need to lyophilize a product, we'd want to work with the manufacturer that can do the upstream, the downstream, and the lyo and fill finish and everything with all the right equipment in place, as well as the analytics, as many as possible.
I'm curious, where would you see your company, let's say 3 to 5 years? Where would you ideally be?
Well, locally in the same place, in the same geography. I'd like to expand our geographic footprint, work more in Europe. I think there's a lot of opportunity and unmet need in Europe. There's certainly a lot of really good people. Also, Australia and Asia Pacific areas are important, I think, for geographic expansion. I like to think globally, like you mentioned earlier, can this be tech transferred south of the equator? And I think we have some opportunity there with our malaria program and some of the other programs that we're developing.
And what does this look like on the cost side? Because often in these geographies, cost is a big issue. Is that comparable to biologics? Better?
Oh, it's certainly better for our product. We can do things much more cost effectively in terms of manufacturing. We don't have the problems of endotoxin like a lot of people have to deal with because we use our cell lines that are relatively endotoxin-free, and we have those all well characterized. So we can have a relatively robust low-tech process that can be used on the benchtop. Another differentiator is with our vaccine products is that we don't use pathogens. A lot of companies are using pathogens, so they require special containment, such as biosafety level 2. They require certain positive air pressure going in and out of the lab and complex filters or a BSL-3. We don't even go there. We use simple non-pathogenic strains and then engineer our products into them that are also non-pathogenic and do not cause disease in people. That's a big differentiator between us and what other people are doing using these technologies.
Let's make this actionable to the smart biotech scientists listening. What's the single piece of advice you would give them about navigating the path you have navigated well from discovery into phase 1? What should they focus on?
Knowing your blind spots. You have to have people around you that can support you and understand what you need to do early on. For example, one of the things we did early on were critical quality attributes in our analytical tests. We put a lot of energy and time into that. I didn't even know what they were, in all honesty. I kind of had a sense, I go, oh, that's what you mean. You mean the analytical tests. And they go, yeah, you need to know batch consistency. You have to know stability. You have to know what is a good product and what is a bad product. So having good consultants and team members that can help you navigate that and design your experiments so that you tick the regulatory boxes, what they're looking for to make it acceptable for human use. You need to do that early on. We had to develop monoclonal antibodies right off the bat. They said you need monoclonals to your target, period. And not only do you need them to your target, you need them to your vesicles. So we did that. So we know how many vesicles we have, how many molecules we have of our product in terms of micrograms, the actual number. So we're able to quantify everything with critical quality attributes. So one of the first things we did was we made our monoclonals and contracted that out and had those delivered. I mean, now people can turn them around pretty quickly. So that was extremely helpful having those in place. And once we had those, then we're able to carry on with the rest of the analytics that were more standard, like osmolality and pH and kind of the basic other parameters, sizing.
Now, circling back to your career path, I'd love you to share what was your biggest learning now as a co-founder and CEO of a company?
Probably you can't do it all by yourself. You rely on your friends and you need to work with people. So for example, going back to critical quality attributes, I met one fellow at a Boy Scout event because we were dads and our boys were in Boy Scouts together and we were talking about immunology, and it turns out he was a CMC guy. So he ended up working with us as a consultant and getting all our critical quality attributes in place from our first team meeting with the NIH. And having him on board was extremely helpful to kind of set the direction for the company and be able to define our process early on about what is a good product. So I think my biggest learning was get as much support as one can and try to do it in a cost-effective manner as much as possible.
Yes. If someone wants to start their company and they have a good technology, they see an unmet medical need, how should they go about to make sure this endeavor is successful?
I think a lot of this effort is being championed by the NIH, or it used to be. And that's where you understand who's the end user, who's going to use the product. So we talk to physicians about, if you had this product, would you want to use it? And if so, how would you use it? So understanding that how it would be used in a patient setting is really important. And understanding the unmet need and how it can complement current existing therapies rather than actually replacing, I think is really important in having that understanding. So they have I think it's called ICOR, is a program that's being championed a lot by the National Science Foundation here in the States and the NIH as well. When you have an early product, understand its use and its implementation in the setting that you expect it to be used in. So talking to physicians, talking to primary care support people, I think is very important. I had that luxury because I had people in my network, and I'm based here in Massachusetts, where you don't have to walk very far before you run into somebody else who's in biotech or high tech.
This has been great, Christopher. What additional questions should I have asked?
My imagination could run wild with that. I think there's a lot of opportunity with this vesicle technology because they can be delivered through the skin. I think in terms of cosmetics and skin diseases, there may be opportunity there. They also can be delivered orally. So like I mentioned earlier, delivering these antibodies orally or other immunotherapeutics orally, I think would be pretty neat to do. All this is going to take clinical development work. And right now we have a lot of ideas that we need to prioritize and just bring one product at a time to the clinic. I think you'll see that in the literature, these vesicles are everywhere. Plants make the vesicles. They're in the oceans. They're used in cell-to-cell communication. Ironically, I was doing HIV research for many years at the University of California in San Francisco. And I worked with the co-discoverer of HIV, Jay Levy. And he published his paper, I think, a few months after Bob Gallo and Luc Montagnier did on the discovery of HIV. And he spent a lot of time on the immunology of it. And he found that T cells can actually block the virus replication through a non-cytotoxic mechanism. And people for years said, oh, no, those are cytotoxic T cells. And he says, no, they're not. They're not cytotoxic. They're like suppressor cells. They suppress the virus from growing. And it turns out it's mediated by little vesicles, could be mediated by vesicles. You can purify the vesicles from CD8 positive T cells, apply them to CD4 positive HIV infected cells, and suppress virus replication. So people thought for years it was a single protein or a handful of proteins of chemokines and cytokines, and that may be true, but a lot of them are packaged in the vesicle. So my point is that it's complicated. There are regulatory RNAs, there's proteins, there's cytokines. Everybody is right in that it is complicated. There is a lot of different proteins, and there's a lot of different regulatory functions going on. And when we break things down, again, to these single molecular interactions, you lose sight of the big picture. And I think working with nanovesicles and engineering them can help us take a step back and look at the big picture and do a deep dive into mechanisms a little bit later. But first, show that they're protective or useful and have their utility in a disease model, and then work out how they're working later on. Almost like a phenotypic screen is what people typically call that.
And to what extent could you use these nanovesicles, for instance, for other applications? Or let's say like this, someone has a drug and wants to deliver differently. Could they potentially use your technology? Is this a business model you're pursuing at all, for instance, to sell your platform?
Yes, we've had some discussions about this. So people that are hampered by delivering therapeutics that are currently doing it with liposomes, they have their own issues with it and drawbacks with liposomes, such as PK and properties. So yes, this is part of our business development strategy, is to do co-development projects or out-license our product for niche applications.
Excellent. We have covered a lot of ground today. What is the most important takeaway from our conversation?
I think keep an open mind. I think Einstein said imagination is more important than knowledge. Things can be worked out. I think it takes a lot of patience. It takes a lot of resources, and it takes a whole village of people in development processes to make this work properly. I think also understanding what the really basic minimal requirements are when it's good enough. We struggled with that a lot. There was a lot of things we wanted to do with our process and our development work, but we have to understand what is going to be good enough from a regulatory point of view. And when you start with that, you can simplify your process and do the basic work that you need to do, and then fill in the rest of it later. I think is a pretty good strategy to keep a company lean and small and to meet its deliverables.
Where can people get a hold of you, learn more about your technology, Christopher?
Well, there's a lot on our website at www.versatope.com. There's a whole list of publications there. There are plenty in the literature. People can do PubMed searches on PubMed Central from NIH. There's a ton of publications on these little vesicles. People are doing all kinds of wild things with them. And to get in touch with me, LinkedIn is perfectly acceptable. I do check LinkedIn, and it's a good tool to network with people.
Excellent. There you have it, Smart Biotech Scientist. I'll leave the links in the show notes. Please reach out to Christopher. And Christopher, thank you very much for sharing your passion, also meeting an unmet medical needs. And it was a pleasure having you on the show today.
Thank you, David. I appreciate it.
That wraps up our conversation with Christopher Locher, from the science behind Versatope's vesicle platform through the manufacturing, regulatory, and analytical challenges of building a first-in-class biologic, to his perspective as a founder and CEO. A lot of ground covered and a lot to take back to your own work. Thank you so much for tuning in today. If this was of value, please leave a review on Apple Podcasts or your favorite platform. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.